Welcome back. We've been looking at how genetic information is transferred from one generation to another. And we know now that DNA plays a key role in the inheritance of genetic information. DNA is kind of like this DVD that I have in my hand. It's nothing more than a means of storing genetic information. Okay, so it's a storage medium. DNA is nothing more than that. So think of that in those terms, like a computer chip. Well, how did that ever get figured out? I want to tell you the stories that are behind this very important discovery in brief. And then we'll look at DNA structure so you can understand something of the nature of how the information is stored in the molecule. Okay, first the history. There is a researcher by the name of Griffith that was looking for the cause of pneumonia in people. And of course, he discerned through experimentation that there was bacteria that was at the bottom of this story. There were two forms of the same species of bacteria, one of which would produce death in laboratory animals, and the other, of course, was not. So where's the difference? So he set out to do some research on that. And it turns out that this transforming material that he referred to from the dead S bacteria could be transferred over to the R bacteria, which made it deadly. Take a look at this diagram here to illustrate the principle further. So we have this live bacteria, which we'll refer to as version S. And when that's injected into laboratory mice, they get pneumonia, and die. Do the same thing with the R bacteria, same species, different variety, however, and the mice survive. How is that? But then if you heat treat the bacteria that killed the first group of mice over here, and you inject them into live mice, they survive. So it's not a protein then. Because protein would be denatured by the heating process of the bacteria. So there's something else going on here that's other than protein, that when you allow this heat-treated bacteria to be mixed in with a live R version of bacteria, there's some transformation that occurs in the R bacteria that produces more dead mice. So how does that work? Well, another researcher by the name of Avery was able to extract and isolate and purify this transforming material that was found by Griffith. And he performed some experiments to find out what its nature was. And it showed that that was actually a nucleic acid that we now know to be deoxyribonucleic acid, DNA. So chemical tests were able to show this. Enzyme tests showed that only DNA degrading enzymes stop the transformation. And when he analyzed 4 different samples, you can notice there is a very close relationship to the amount of nitrogen, the amount of phosphorus, around 9%, and then the ratio of these two was about 1 to 1.7. In each case, which is going to show that their structures are similar. So Hershey and Chase were able to confirm that it was DNA, that is the genetic material, that carried over this information. They did this by studying particular viruses that infect bacteria, known as bacteriophages. So they tagged this viral DNA with a radioactive phosphorus. And so, that was the material that was transforming these bacteria. And they also were able to tag viral proteins with radioactive sulfur, because that's an important constituent of many proteins. It was the tagged DNA that was found inside the bacteria, while the proteins remained outside. So obviously there was a carrier here that carried the bacterial DNA from one species to the other. So we know that now DNA plays an important role in carrying this genetic information. And it's so important that it was responsible for producing the death in the original mice. Well, What is the structure of DNA? We'll get into that soon. Key idea that we want to keep in mind at this point is that the DNA structure is fundamentally the same in nearly all organisms. I think there's under 20 organisms that use a different structure. And they're all made up of basically 4 nucleotides. So we have a long chain of these monomers, these smaller molecules that make up very long, large molecules of DNA. Every one of those 4 nucleotides has these 3 parts that you see pictured here. A phosphate group, which is phosphorus and 4 oxygen atoms. A 4-carbon sugar known as deoxyribose, and a nitrogen-containing base. And this is the part that's going to change from one of the 4 types of nucleotides to the others. All right, so 3 parts, okay? And here are the 4 different versions of these nucleotides. Thymine, we just represent that by the letter T. Cytosine, we represent by the letter C. Adenine, which is a little bit larger, which we refer to as a purine because it's got a double ring structure, and then one other purine, which is guanine. So A, T, C, and G. These are the letters that we use to abbreviate references to these nucleotides. Okay, so there's only 4. Okay, the overall structure, the 3-dimensional structure, was first realized by Watson and Crick, 2 researchers, back in the 1950s. Okay, 3-dimensional structure of these building blocks. And as you can see in this photograph here, they realized that the structure itself was a twisted ladder known as a double helix made up of the sugar phosphate backbone that we saw on the outside of the nucleotides. On the inside were those nitrogen bases that where the genetic information is actually stored. That's on the inside. And this work is based upon the work of Franklin and Erwin Chargaff. And they did a lot of research that had to do with X-ray crystallography, which is shooting X-rays through crystals, in this case of DNA. And they were able to determine the even width of the DNA molecule as you go down the spine of that it's an even width. They also found and coded a very important rule that you can see here, that the amount of adenine and thymine are proportionate to each other, and the amount of cytosine and guanine are proportionate to each other. So that's come to be known as Chargaff's rule. And it turns out further, there's a reason why that rule holds true. And the reason why is because every adenine is going to be pairing with the thymine, and every cytosine is going to pair with a guanine that you see pictured right here. So inside this double helix, this twisted ladder structure, we see actually stored and protected what we now refer to as the genetic code. These nucleotides here really form the basis of a 4-letter language, similar to the alphabet that we use in making our words. We use 26 characters. The language of DNA only uses 4, but it works in a similar way, because the order of these nucleotide pairs is how the information is stored. And we're going to look at this closer because how this is held together. The single ring structure, the pyrimidine, and is going to bind with a purine, the double ring structure, to keep the width uniform. So the overall structure of the double helix remains to have the same diameter throughout. The backbone, where the phosphates and these sugar molecules is going to be held together by covalent bonds. You see it labeled here. And far weaker bonds hold the pyrimidines and the purines together going down the middle. So I'd like you to liken this molecule in your mind to something of a zipper that you would find on a sweatshirt or a sweater or a jacket that you have. These bonds are much weaker, which means that the molecule can unzip right down the middle when it comes time for DNA either to be opened so that information can be unlocked and read, or so the molecule can split and be copied and make multiple copies of the same genetic information flawlessly. Okay, so we've looked at the experiments that led up to the discovery of the structure of DNA, and we've considered something of the structure of DNA itself. It's made up of these nucleotides. 4 letters. A binds to T, C binds to G. Base pairing rules. Thanks for watching. Bye-bye.